Hi everyone, my name is Victor and I'm going to present to you the research diversity of the genus Aegla in a highly endemic region with guidance for Professor Sandro Santos and Professor Marlise Bartholomei-Santos. So the genus Aegla is composed by 93 species, but this number could be much higher due to the crypticity found in Aegla paulensis, Aegla longirostri, and Aegla uruguayana. They inhabit freshwater environments with good physical-chemical conditions, and they are dispersed through Argentina, Bolivia, Chile, Paraguay, Uruguay, and mainly Brazil. Here in the southern region of Brazil, we have a priority region for Aegla conservation. It's due to the high species richness, phylogenetic diversity, elevated endemism, and threats to habitat found in that area. And this region has also protected areas that could beneficiate the Aegla species. So our main objective here is to unveil the actual Aegla diversity in the priority region for conservation. For that, we used 33 sampling sites distributed in a 9,000 square kilometers area, inside and in the outside of 4 large protected areas in the southern region of Brazil. We have 3 sampling campaigns, and in each one of them, we had captured the specimens using active search. Then we performed the morphological identification of these specimens, the DNA extraction, and the PCR of the mitochondrial gene CO1. After the sequencing, all the sequences that we retrieved were treated in staging software, checked for the quality, and then aligned in Mega software, and also checked for quality. For the phylogenetic analysis, or 151 sequences plus 132 Aegla sequences from GenBank were used to perform a Bayesian analysis using Bayesian method implemented in BEAST 1.8. And for the species delimitation, we performed 2 types of delimitations. The first one is the general mixed yield coalescent, and the second one, the assemble species by automatically partitioning the asset. Our results then. For the species delimitation, we have retrieved 8 known species that had grouped with the well-described species as in Aegla camargoi, Aegla inconspicua, Aegla leptodactyla, and Aegla serrana, as you can see in the right side. And together with that, we have retrieved Aegla franciscana, Aegla Leach, Aegla oblata, Aegla spinosa. They are also confirmed by the morphology. But surprisingly, we have found possible 12 new species divided into 3 cryptic complexes. 8 in Aegla jarai, as you can see in the image from the right side. As an example here, we have the jarai complex. When it's divided itself in 8 parts, or it means possible 8 new species, and 3 in Aegla franciscana and 1 in Aegla camargoi. We have found another 11 new taxonomic units that hasn't grouped with no sequences from species of GenBank, all right? So what's the implication of this for the conservation? Most part of the species found in our study are restricted to only 1 or a few sites. It also found mainly in outside protected areas, in regions with high anthropic pressures. So given these factors, describing these species is of a great concern in order to ensure specific conservation and management efforts. And other conclusions is to integrate traditional taxonomy with genetic procedures should be the way to understand the real diversity of the group. And our results indicate that the diversity of the genus is largely unknown and reinforces the need for more studies and conservation efforts for this taxon that is very important to the aquatic environment. I would like to thank all these people here, especially the Professor Georgina, for delineating the job. And here is my email if you have some questions or another subject to discuss. And thank you for your attention.